questions before I dive into 4, 6, 8, 10, 11? There's 5 people missing today. All right. No questions on what we have been talking about up to this point. We've been talking about the various pathologies associated with eating gliadin, which is one of the components of gluten. And I was telling you that there's a variety of ways that gliadin can affect an individual. Some of these are genetically mediated, meaning a person is sort of born with the sensitivity. In particular, there's 2 ways that this happens. The most common genetic predisposition is having a specific human lymphocyte gene, an HLA gene, that human lymphocyte antigen gene that is expressed on your immune cells, your white blood cells, that cross-reacts with part of the alpha gliadin. And that means that the HLA antigen is going to be part of an inflammatory cascade in the body through mediated by the immune system, which leads to the sort of inflammation that you'll see in celiac disease. There are also some non-HLA genes that can lead to sensitivity in the alpha gliadin. On the other hand, there are all these environmental risk factors. For example, if between the ages of 4 months to 6 months, 8 months maybe, you expose your infant to a high degree of gluten all at once, right? That is correlated with an increased sensitivity to gluten when the child gets older. Another environmental risk factor is if a child has chronic rotavirus infection. So rotavirus is basically it's a viral infection of the lower GI tract, so basically diarrhea. If a child comes in contact with with rotavirus, particularly if they have a recurrent infection with rotavirus, that tends to lead to an increased sensitivity to alpha gliadin. Why is that? Well, because one of the things that rotavirus does is it increases intestinal permeability. So the enterocytes, or the epithelial cells that cover the inside of your gut, become more permeable if you've had a chronic rotavirus infection, and because of that, the person just is exposed to or absorbs more undenatured gliadin in those instances. And a third environmental risk factor is socioeconomics. So there's this the sort of hygiene hypothesis that the increased incidence of all types of allergy, not just allergy to wheat, are due to the fact that in sort of modern society, we live in these kind of hyper-hygienic environments where people are not, your immune system is not sort of put to the use for which evolution intended it, right? We're not really exposed to the range of bacteria, the range of other pathogens that our body has the machinery to cope with. And because of that, all this machinery that had an intended purpose doesn't ever get the chance to execute that purpose, instead looks for other reasons to do that, and often that manifests itself in terms of allergy. So these immune responses to things that aren't actually pathogens, and thus the immune system gets trained with time to respond to things that it shouldn't be responding to. Okay, and so this is not exclusive to wheat allergy. This is manifest in all kinds of different allergies. It can be atopic dermatitis. It can be atopic rhinitis or hay fever. Atopic dermatitis is like eczema. Asthma is another atopic disorder. Wheat allergy or other inflammatory bowel disorders are another manifestation of that. So this is an instance where higher socioeconomic status actually puts you at greater risk rather than lesser risk of this problem, meaning hygiene. Right? And I don't know. I guess I could have put some slides in here. I didn't. Kind of so contemporary pediatric epidemiologists or pediatric immunologists suggest some things that may be a little bit surprising to at least my generation and certainly my parents' generation. I don't know how you guys feel about it, but they say things like when kids are little, it's not just okay, but fine and encouraged for them to pick their nose and eat it. Yeah, I know. Yeah. But the point being that it's giving them a baseline of exposure that sort of trains their immune system in a healthy way. But we live in a different sort of world. Did I talk about fecal replacement therapy in this class yet?
Is that taking someone else's poop and giving it, putting it nicely to someone else?
Mm-hmm. Yep, that's right. So taking a healthy fecal sample, a stool sample, isolating the bacteria, the healthy bacteria, the gut microbiota in it, and then administering it in one way or another to an individual. Typically, it's most effective against people with C. difficile infections in their gut who have really screwed up gut microbiota, often because they've had a lot of antibiotics that have thrown their system off. But so anyways, we live in this, my point is not to gross you out, but it's to let you know that we do live in this era of sort of a new and different understanding. Would you close the door just to a new and different understanding of our human health, our immune system, our baseline endocrinology and the hormones in our body, and its relationship to the microbial and viral flora that are out there, right? That are not just out there, but are on you and around you, and that you come into contact with. So another thing, this woman I was listening to speak about this pediatric epidemiologist, that like food that falls on the floor, don't eat that. Well, I mean, you have to. She said, actually, it's not always that bad to give yourself exposure to a little bit of the world out there. So, anyways, that's an environmental risk factor for sensitivity to gluten.
Mm-hmm. Is that only in kids, or can you still like train your immune system once you're older?
So your immune system is always adapting to its environment. Yeah, if you want to start picking your nose and eating it, it's probably not going to hurt you. Yeah, so.
Just time to start now.
I'm not necessarily advocating it. I'm just telling you what other people say. Yes.
At like what point?
Yeah. Right. I mean, I don't know if I'm going to be able to draw a clear line for you like that. I don't know. I think it becomes unhealthy when you put something in your mouth that makes you sick. What that point is, I don't know. I would say use common sense. humans have an immune system because over the ages we descended from creatures that didn't live in buildings. We've created buildings. We lived on Earth for eons, eating food, coming in contact with organisms that are what would be considered of questionable, quality to a modern person. I think keeping that in mind and like recognizing that we don't need to live in a sterile environment and that living in a sterile environment is not healthy, in fact, for us. But at the same time, finding your comfort zone with what you are willing to tolerate and do. And I think not succumbing to our neurosis in one hand, but also not becoming I'm not telling you guys that we should be, living like animals or something like that. I don't know what the answer to your question is, frankly. I think that's something that is, you have to answer that for yourself. It becomes unhealthy when it becomes an obsession. It becomes unhealthy when it leads to, quantifiable measures of physical health going down. Whatever that means. Maybe you eat something and you get sick from it. You get food poisoning. I don't know. Exactly. It's a very hypothetical question. Make sense? Yeah. Any other questions about that? Well, anyways, I have this, I don't know, I like putting pictures on my slide, and I didn't put pictures with those risk factors, but I did put this map of the gliadin molecule. And it has the different little molecular motifs, the regions of this protein that elicit different responses. So this little red loop down here has some amount of epithelial cytotoxicity. That means that red loop causes, in certain individuals, can cause epithelial cells to die. Cytotoxic means toxic to cells. And specifically the cells of the intestinal lumen, right? So not in all individuals, in individuals that have this predisposition to it. So they've identified that as the region of epithelial cytotoxicity. The part that's yellow has been recognized to have immune modulatory functions. So it can change the functioning of cells in your immune system. The blue regions have an effect on gut permeability. So this is the ability of whatever proteins or organisms to pass through that epithelial layer that protects the inside of the gut, the lumen of the gut. And then lastly, there is this tail region, this C-terminal tail region that stimulates the production of interleukin 8. So interleukin 8, in celiac patients, interleukin 8 is a hormone. The interleukins are hormones that they can do a number of things. They can be pro-inflammatory or anti-inflammatory, but they're things that cause a cell to go through some action, some sort of automated process, and that hormone will turn on that process. Interleukin 8 tends to be a pro-inflammatory interleukin. So it starts the inflammatory process. It's like sending out a light, a beacon to the immune system in that region to send more immune cells into the area. Yeah, that little tail causes the production of interleukin 8. All right. So that's the sort of the pathology that is behind this gluten-free market. Now, gluten-free is the market itself is probably scaled beyond the actual prevalence of the pathology out in the world. If you look at just the market share of gluten-free, it's been exploding. It's really been exploding, and driven by the increased diagnosis of gluten sensitivity, but also I don't know. It has become a bit of a fad, dependent upon people's misunderstanding of what gluten even is, and whether they're sensitive to it or not. Some people just feel like it must be bad, and I want to eliminate it from my diet. And so that just because they want to be healthy without fully understanding what it is that they're trying to do, it has become a giant business. So this year, $7.42 billion, and that's set to reach $8.45 billion by 2025. That's a huge amount of money in the food market. You can see that a lot of it is being driven by bakery products. Of course, is the largest share of them. Bakery products obviously typically have gluten in them, and breads, etc. And then the dairy alternatives, some others, prepared foods, can have a big chunk of that market. So what does this come down to in terms of what people actually spend? So this is just a chart showing the average weekly grocery bill for Americans, adult Americans. And this infographic, I cut the source out. I don't remember the year, but I think it's within the last 2 years that these statistics apply. So, yeah, it's a lot of money. A regular, an average individual spends about a little over $60 a week, about $62 a week on groceries. And if you are trying to be gluten-free, that adds about 25% to your weekly grocery bill to be able to purchase food that is gluten-free. Clearly, organic, we recognize that organic food is more expensive. It's a more, the economics of growing organic food makes it so that the price point has to be considerably higher than non-organic food, and that's about $100, about 40% higher. But gluten-free is not an insignificant amount of financial burden to the people who have to be gluten-free. So, yeah. What's that?
Why can people?
They can't eat all rice, and people with severe celiac probably shouldn't eat any rice. But there are types of rice that are low gluten, very low gluten, and so if you are not an acute celiac, then you can eat some rice. Does that make sense? Okay, so anyways, are there questions about any of this? This is where I'd wanted to get last time. This is the end of my protein spiel. No? Okay. So now we are going to switch gears and talk about a little bit of carbohydrates. I'm not going to be able to get through this should be lecture 5 or topic 5. Sorry. Update that. I'm not going to be able to get through all of the carbohydrate material I want to talk about before the midterm, but I'll talk about some today and maybe a little bit on Wednesday. But before I do that, what I would actually like on Wednesday is if you guys have questions that you would like to ask me review over any of the material that we've had, you come to class. I'm not going to prepare a review, but I will review things if you come to me with questions. Does that make sense? Right. If nobody comes with questions, I'll just keep talking about carbohydrates. Yeah.
I have a question right now. Like, how much is going to be on the midterms? So just so I can
25 multiple choice questions and an essay.
Essay. Is it a specialist?
Very special. I don't know what special means. It's going to be an essay. I haven't written it yet, but it's going to be an essay looking at
one of these topics.
I'm probably not going to dive maybe I haven't written it, but I'm imagining what I'm thinking about with the essay is something where I'm getting you to think more broadly about the themes that we've been covering this semester, and drawing on some examples of stuff that we've talked about, so you can apply some of your knowledge but speak more discursively about the topic. Does that make sense?
Yeah.
Yeah. I wish I could tell you what the question was, but I can't. All right. Any other questions? Bring that stuff on Wednesday. Yeah, we'll get into it if you guys want, or not, whatever you want. Okay, so the biomolecules. We're right here on the biomolecules, working our way up to the baby who has the whole world in his hand up there. And we've talked about proteins. Next is carbohydrates, and then we'll get to some lipids after the break. And then after that, I feel like I've covered all of the primary concepts, and we'll start exploring a little bit. I'll talk about metabolism and taste, and maybe some chemical changes that happen to food, whatever. We'll see what we can get to. But proteins, carbs, and lipids we got to do. Carbohydrates. I actually, my dissertation was in carbohydrates. I identify as a carbohydrate biophysicist. So this is a topic that's very close to my heart. A lot of people, I don't know, maybe I've said it in here already. I'm a fount of facts, little factoids about carbohydrates, but 85% of the dry biomass on Earth, that is 85% of everything that is alive on Earth, is made up of carbohydrates. And the vast majority of that 85%, like 83%, is actually some sort of polymer of glucose. So by and large, Earth is a big ball of glucose. And we'll see what that means. And there are some reasons for that. There are a number of reasons for that. I'm not going to be able to give you all of them here because we don't have time to take that deep a dive. But one of the things I do want you to understand about carbohydrates is, so, you talk in the media, and carbohydrates currently are pretty demonized, right? They get demonized. It's like carbs. People don't keep me away from the carbs. I'm an addict. That kind of thing. In fact, carbohydrates are far more. The function of carbohydrates is far more varied, diverse, and pervasive than people appreciate. And beyond that, carbohydrates are really not just most of life on Earth, but they were sort of the beginning of life on Earth, or at least the beginning of the chemical economy that led to life, if you want to think of it like that. When I say chemical economy, I'm talking about the self-sustaining chemical interconversions, the cyclic chemical reactions that happened in the prebiotic realm when when Earth was entirely sterile. There was nothing alive. There was no plants. There was nothing but water and rock. There was a point when Earth was just water and rock. It's hard to imagine, but it's true. And at that point, the sun was shining down, and there were various redox conditions on Earth, meaning balance between oxidation reactions with oxygen and reduction reactions. These redox reactions were catalyzed and driven. Certain categories of them were catalyzed and driven by the presence of iron. There's a lot of iron on Earth. Maybe, for example, why does the Earth have a magnetic pole? What do you think?
That is because of the meteorites, the metal meteorites that hit the Earth a long time ago and sank all the way to the crust. I mean, it sank past the crust into like the mantle.
You're getting close. I don't think it had anything to do with meteorites, but the whole core of the Earth, the magma core of the Earth, is entirely molten iron, right? So the whole inside of the Earth is a mass of molten iron, which is why we have a magnetic pole. And there's lots of iron on the crust as well. And it's this iron that can have what are called different oxidation states. And, without getting deep into the chemistry, the oxidation state of a metal like iron is simply a reflection of the electronic structure in what's called the valence shell, those outer electrons. And that electronic structure, the valence electron structure in iron, the different oxidation states is what you call that, can drive the fixation of carbon dioxide that's in the atmosphere into larger carbon-containing molecules, and that's what we see here. So we have Fe2 plus and carbon dioxide and a little bit of acid, okay, some acid in the broth, the primordial broth, absorbs some energy in the form of UV light. That's the 400 nanometer light, the HV light that I have on the left, and out pops iron 3 plus with a changed oxidation state, and then the release of very simple carbohydrates. All right, that are very simple organic molecules that lead to carbohydrates. So we have the fixation of carbon dioxide to acetaldehyde, which then leads to carbohydrates. So let's look at the name carbohydrate. I guess, let's go back here. When I say the word carbohydrate, what do you feel like you could tell me about what a carbohydrate is? Just from what you know. Yeah, what?
It's energy, and not in its purest form of ATP, but it is.
Okay, energy, really important. You need it. What else? What else do you guys know about carbohydrate? Yeah. Oh, good. Yes, that's right. It's carbon and water, and we'll see exactly what that means in a minute. Alex, what are you going to say? Okay, yeah. The name itself tells you something about it. They called carbohydrates early chemists, really. They weren't really nutritional scientists at the time, but early chemists gave them that name because it was when they were trying to analyze carbohydrates, they found that they all had this elemental ratio, this empirical formula, this elemental ratio that had carbon and hydrogen and oxygen in a 1 to 2 to 1 ratio with one another. Okay, so all carbohydrates are going to have a ratio of carbon to hydrogen to oxygen in a 1 to 2 to 1 ratio. So that's some sort of, the way they saw it was some sort of hydrated carbon that had water on it is the way they initially understood carbohydrates. Now, it's not a bad description, but we're going to make it much more nuanced as we move forward here. So when you're talking about sugars, carbohydrates, another word that you'll hear is saccharide. Saccharide just means a sugar. A monosaccharide is synonymous with the term simple sugar. So a simple sugar is a single carbohydrate molecule. And we'll see that we're going to be able to link them up because these are all biopolymers, right? And we can link these sugars into long chains, which is what starch is, for example, or cellulose, wood fiber, and et cetera, et cetera. We'll see examples of that. But the simple sugars are these monosaccharides. And on the left, you see glycerol, also sometimes called glycerin. Glycerol is a 3-carbon molecule. You see there's 3 carbons in a chain, and then each of the carbons is attached to an OH group. Okay, so an alcohol group. We've run into these OH groups before. They're alcohol groups. Glycerol has a carbon chain, each of the carbons attached to an OH group. Glycerol is not a carbohydrate. It is a relative of carbohydrates. If you were to look at the formula of glycerol, we would have C3, H, what is that? 3, 4, 5, 8. C3H8O3. That's not 1 to 2 to 1, is it? No, it's not a carbohydrate. But this molecule on the other side is called glyceraldehyde. And instead of the bottom 2 carbons are just like it is in glycerol, except that last carbon has been oxidized. It has increased the number of bonds that carbon has to oxygen. All right? That specific chemical group there is what we call an aldehyde. That C double bond O is a carbonyl group, and when it's on the end of a carbon chain, we call it an aldehyde. All right? So glyceraldehyde has a C3 H6. If you count up those hydrogens, there's 6 of them in there, and there's 3 oxygens. So that is a carbohydrate. It's C3H6O3. And you can see in that top, yeah. Yeah. The aldehyde is simply that carbon, the terminal carbon, is double bonded to an oxygen rather than single bonded to an oxygen, which in turn would be bonded to a hydrogen. Okay, so like in glycerol, they have all these hydroxyl groups, these OH groups, right, that have single bonds to the carbon. But in a sugar, one of those carbons, so all of them have hydroxyl groups on them, except one, which has a double bond O attached to it. And if that double bond O, that carbonyl group, is on the end of a carbon chain, we call it an aldehyde. And if it's in the middle of a carbon chain, we call it a ketone. And we'll see examples of that. So, for example, glucose is an aldehyde. It's in fact an aldose. The suffix "ose" refers to sugars. So glucose is an aldose. And fructose is actually a ketose because the double bond O is not at the end of the chain. We'll see that in a moment. But this is the simplest sugar, so to speak, glyceraldehyde. And if you were to taste glyceraldehyde, it has a sweet flavor, not what I would call especially pleasant, but not glucose or anything. So we can then begin to build. So glyceraldehyde is this top molecule here in this tree, but we can begin to build a tree, a sort of network of carbohydrates by simply adding one carbon with a hydroxyl group attached to it in the chain and just lengthening that chain. All right, so glyceraldehyde is the only triose, meaning 3-carbon sugar. There are 2 tetroses, erythrose and threose. So if you look at erythrose and threose, you'll notice that second carbon in the chain, not the one boxed in red, but the second carbon in the chain, they have different orientations. All right, they're one's facing one way, and in the erythrose, it's off to the right, whereas in threose, it's pointing off to the left. And we can continue to extend these chains. So pentoses, there are 4 permutations that you can have. And then in the hexoses, the 6-carbon chain sugars, there are actually 8 different permutations of what's called configuration about each of the carbons in that chain. I'm going to explain the stereochemistry a bit in a moment. But I just wanted you to see how we could build up these chains and make different individual sugars that can be used in one way or the other by the body. So all of these sugars at a particular level, say all of the hexoses, for example, they're all what are called isomers of one another. So an isomer is a molecule that has the same number of atoms in it. All right. So for example, all those hexoses are C6H12O6, but they're connected in slightly different ways. All right. That's what makes them isomers. Any molecule that has the empirical formula C6H12O6, that's going to be those are all isomers. Does that make sense? Okay, so all of those sugars in each of these different levels of the tree are isomers with one another. Here, I have outlined, not all these sugars are made alike, so to speak, in terms of biology. I've outlined the ones that you will commonly run across. You can find the other ones out there, but in nature, some of them you won't find in nature. But these are the ones that are commonly found in nature. So glyceraldehyde is an important part of central metabolism in every cell in your body. Ribose is it's the ribo in deoxyribonucleic acid. Ribose is part of RNA and DNA. And you've all heard of glucose, that's blood sugar. Mannose is a sugar that is found in complex saccharides in the body that have sort of higher level functions, not for energy, like you were saying about carbohydrates. Most people, when they think carbs, they think energy, but carbohydrates do a lot more than just give us energy. And galactose is similar. We had a little bit of a taste, so to speak, of galactose in lab on Friday, I think. I put lactose, which is mother's milk, up there, and that was a disaccharide of galactose and glucose. So galactose is kind of like glucose in that it serves some other more obscure, higher level functions. All right, so to really get a sense of what I am talking about when I'm just switching molecules back and forth, I want you guys to be able to think about molecules in space just a little bit. All right. So what do we have here? We've got three groups of 4, I suppose. So what I would like you to do is get in a group of like 4 or 5 and just take a minute and look at these 2 things that I'm handing you. I'm going to get the full review one here in just a second. I'd like you to take a moment, circle up, and look at these things I'm handing you and tell me what you think the relationship between them is. What is the relationship? And so now I have, the way I've labeled them, you can see that there are 4, there's a central atom, right? And we're going to call that carbon. And then we're going to call each of the arms that are sticking out from it some sort of atom that's different from the other one. All right? Each of them is different in identity. And the ones I have on the slide up there, it's a different molecular modeling set, but that doesn't matter. So why don't you guys, you 4, just give me 2 seconds, but the rest of you, just circle up and look at them, toss them around in your hands, pass them around, and see if you can tell me, come up with a way of describing the molecular relationship between those things.
So if you're able to like simulate on computers the interaction.
Yeah. Absolutely. That was part of my research.
Tricking us.
On Earth, yep. I mean, it was, I don't know, most important thing. It's like a lot of different steps that led to it, but yeah, carbohydrates were. What's that? Yep. The color of the arm. So you'll see there's a what I'm calling a red arm, a white arm, a black arm, and a gray arm. All right, you see what I mean by that? And are the two things I handed you identical? If they're not identical, how would you describe the relationship between the two of them? I'll make some more here for people. Who wants another set? All right. Okay.
You have another one. Well, the color matters. Color matters. I think color matters.
Okay, I'm gonna give you guys another one too. I'm out of colored balls, but that doesn't matter. So you can still call these arms different because there's like, do you think the barrel and the
No, usually in the whole like. I know this because I asked my chemistry. Usually, what is?
Are the balls supposed to be like a pattern? And then the arms supposed to be just
bonds? That's right.
So are these two bonds like
Usually,
usually?
I usually.
Yeah. So imagine this is an atom. This is a different atom. Third atom. Much
Middle
little
one. Are we supposed
I
to
just
assume
don't
that there's a
have
middle
my
atom
nice
here?
atom table.
So there's supposed to be something.
Yeah. There's
Pretty
atoms
sure
there. I
that
just
we
don't.
should pretend carbon.
Just don't have a ball there. Are they the same?
You're an
Okay.
isomer.
So let's take another minute. You guys saw. They certainly are isomers. What? Can you give me more?
You want the relationship
more
between?
description?
Do you want the relation?
Okay. Well, what's different? Give me some language. Language.
Of the bonds?
Yeah.
Yeah. Carbon was created that double.
Because I just have this red in the middle and the black over there. Special relationship. How about it? You guys have any coming up with the language? They are isomer. But there's a further special relationship because couldn't this red atom be at the center and have this carbon out here, and they would still be all the same atoms.
Okay.
But there's a special relationship that I'm trying to get at here in this.
Have we talked about it?
I haven't given you the name. I'm not expecting you to give me the name, but I want a descriptive language. If you were going to describe to somebody, you say, okay, I'm going to give you 5 atoms. You're going to connect them with one atom in the center. I want you to give me the language that you would use to tell somebody, instead of saying, tell them how they were connected, in a succinct way. I like what you're doing there, the way you're holding those. Yeah, what do you think? Yes, they're mirror images. They're mirror images of one another. let me see here if I can do this. Can you, so you are about 85% of the way where I would, aspirationally hope you to get. What else can you say? Can you guys see that they're mirror images of one another? So here, I'm about to give you this. This model here. These are hard to get together. I don't like these. How is that set that you have different than this set? Here, I'll give you guys a set like that too. So,
Wait, now it's a double. Should be that big bond in the middle. I think it's a big bond beats double.
what is it? What else about the mirror imaging? Yeah.
The one you just handed has a double bond, that thick.
No, I didn't give you any double bonds. Nope.
Okay.
Double bonds are these in this molecular kit.
Oh.
No, those are all single bonds. They're all single bonds. What is different about what I handed you?
So, this one. That's gonna be
Are they mirror images of one another? Are these things mirror images of one another? Yeah, they are. Okay. So what's different about what I just handed you from the ones that we have? He's right, they're mirror images, but there's one other component that makes them special. What's that? No. I'm sorry, that's not you. It's my frustration with the cheapness of the kits that I have. None of them can connect. Don't imagine that they can connect. Because the ends of each of the arms, we'll assume they're all atoms on there. There are no open bonds. So as you're messing with them, think about the mapping that you can do. All right, can you map the first ones that I gave you with the colored balls on the end? Can you map one of them onto the other? Can you?
Yeah.
Is that a mapping? So you have the white one mapped, the black one mapped, and the red one mapped, but the grays aren't mapped. One gray tip is not where the other gray tip is. Can you map it? Yeah. Okay, we'll take the two that I gave you, the second set. You can map. What I'm getting at is what you see here when you have a carbon in the middle of this tetrahedral structure. They become non-superimposable mirror images. That's the thing. They're mirror images, but non-superimposable. That is at the key. That's at the center of carbohydrate function and structures. This idea that all of these hydroxyl groups going down the carbon chain form these non-superimposable or chiral centers. So we'll pick up with this idea next time, and we'll talk about my academic great-grandfather, this guy here, Louis Fischer. Emil Fischer. Huh? Academic. So the guy I got my PhD from got his PhD from a guy that got his PhD from this guy.